Modifications of the PCR technique. Nested PCR. Nested PCR increase the specificity of DNA amplification by reducing non-specific amplification of DNA. Two sets of primers are being used in two successive PCR reactions. In the first reaction, one pair of primers are used to generate DNA products, which besides the intended target, may still consist of non-specifically amplified DNA fragments. The products are then used in a secondary PCR reaction with a set of primers whose binding sites are just downstream of the first primer or nested between the original set of primers. So, similar sites are in the first reaction but are completely within the DNA target fragment. Then asymmetric PCR. Asymmetric PCR is used to preferentially amplify one strand of the original DNA more than the other. It finds use in some types of sequencing and hybridization probing where having only one of the two complementary strands is required. PCR is carried out as usual but with a great excess of primers for the chosen strand. Then RT-PCR or reverse transcription PCR, which you might have already learnt because of this COVID situation. We have heard about this RT-PCR so many times. So RT-PCR, reverse transcription PCR, is a method used to amplify, isolate or identify a known sequence from a cellular or tissue RNA. To amplify, isolate and identify a known sequence from a cellular or tissue RNA. The PCR reaction is preceded by a reaction using reverse transcriptase to convert RNA to cDNA. Then hot start PCR. Mixing of all PCR reagents prior to an initial heat denaturation step allows more opportunity for non-specific binding of primer sequence. Hot start PCR is a technique that reduces non-specific amplification during the initial setup stages of the PCR. A technique may be performed manually by simply heating the reaction components briefly at the melting temperature 95 degrees Celsius before adding the polymerase. Then quantitative PCR. Quantitative PCR is used to measure the quantity of a PCR product. It is the method of choice to quantitatively measure starting amounts of DNA, cDNA and RNA. Quantitative PCR is commonly used to determine whether a DNA sequence is present in a sample and the number of its copies in the sample. The method which currently the highest level of accuracy is quantitative real-time PCR. It is often confusingly known as RT-PCR, real-time PCR, but the real one is reverse transcription PCR, which we learned earlier. Real-time PCR is used to quantify gene expression and also to screen for mutations and single nucleotide polymorphisms. Then touchdown PCR. Touchdown PCR is a method used to increase specificity without compromising yield. The principle is to initiate synthesis at very high annealing temperature, which permit only perfectly matched primer template hybrids to form. The annealing temperature is dropped in a stepwise fashion with each cycle. Once copies of the target sequence have begun to accumulate over the first few cycles, high temperature annealing becomes much less critical for specificity as it is previous products which form the major template. These products are unlikely to have any sites for mispriming. The benefit of decreasing the annealing temperature is to increase the probability of stable primer target interaction. Then RACE, rapid amplification of cDNA ends. RACE is a PCR based method for locating precise start and end points of gene transcripts. First step in this method is conversion of mRNA into cDNA with reverse transcriptase. One of the primer used during reverse transcription is specific for an internal region close to beginning 5' end of the gene under study. Since only a small segment of mRNA is copied, the cDNA also generated will correspond exactly with the start of the mRNA. After synthesis of above cDNA, a short poly A tail is attached to its 3' end and is subjected to normal PCR. The second primer will anneal to this poly A sequence and converts the single stranded cDNA into a double stranded molecule, which is subsequently amplified as the PCR proceeds. The sequence of the final PCR product will reveal precise position of the start or 5' end of the transcript. This is known as 5' race, so called because it results in amplification of the 5' end of the starting RNA. Recombinant DNA technology and biotechnology: primer, internal annealing, then synthesis of cDNA, then synthesis of poly A tail using terminal transferase, denaturation and annealing of primer, extension, 5' race. An internal primer is used to prime synthesis from an mRNA template. A poly A is added to the 3' end of the cDNA using terminal transferase. Second strand synthesis is prime using second primer. Then in 3' race, for isolation of 3' end of an RNA, mRNA is first reverse transcribed to cDNA. PCR amplification of the cDNA is then carried out using a primer specific to non-sequence in the mRNA understudy, close to its 3' end, and an oligo U primer. Nested PCR is then performed to improve specificity. These 5' and 3' race products can then be used to isolate full-length cDNAs in a few days compared to weeks required for screening cDNA libraries. Then random amplified polymorphic DNAs, RAPD. Random amplified polymorphic DNAs, RAPD stands for random amplification of polymorphic DNA. It is a type of PCR, but the segments of DNA that are amplified are random. Unlike traditional PCR analysis, RAPD does not require any specific knowledge of the DNA sequence of the target organism. In RAPD, several arbitrary short primers, 8 to 12 nucleotides, and a large template of genomic DNA are used. No knowledge of the DNA sequence for the targeted gene is required, as the primers will bind somewhere in the sequence, but it is not certain exactly where. This makes the method popular for comparing the DNA of biological systems that have not had the attention of the scientific community, or in a system in which relatively new DNA sequences are compared. It is not suitable for forming a DNA data bank. Due to the fact that it relies on a large intact DNA template sequence, it has some limitations in the use of degraded DNA samples. Its resolving power is much lower than targeted species-specific DNA comparison methods, such as short tandem repeats. In recent years, RAPD is used to characterize and trace the phylogeny of diverse plant and animal species. Properties of RFLP and RAPD. Distribution: RFLP ubiquitous, but RAPD ubiquitous. Level of polymorphism is high in RFLP, high in RAPD. Detect allelic variant, yes, but in RAPD it's not possible. Number of loci detected 1 to 3, 1 to 10 in RAPD. Then detectable in all tissues, yes, same in case of both of this. Then reliability high, intermediate or high in case of RAPD. Then quality of DNA required, relatively pure, but RAPD crude. Then quantity of DNA required 2 to 10 mg in RFLP and 10 to 15 in RAPD. Use radioisotopes, yes, but in RAPD it's not used. Type of probes required: species specific low copy gDNA or cDNA clones. Random 9 to 10 mer oligonucleotides in RAPD. Next, amplified fragment length polymorphism or AFLP. AFLP is a hybrid of RFLP and RAPD techniques. Genomic DNA is cut with restriction enzymes, as in RFLP. Typically, two different restriction enzymes are used. So, AFLP is a hybrid of RFLP and RAPD techniques. Genomic DNA is cut with restriction enzymes, as in RFLP. Typically, two different restriction enzymes are used. The idea is to produce a large number of fragments. Some of the fragments are selectively amplified with PCR using random primers as an RAPD. The primers are not really random, however. Some oligonucleotide adapters, these are complementary to the restriction sites of 25 to 30 BP, are ligated to the restricted DNA fragments. The primers are complementary to these adapters. However, the primers vary at their 3' end such that they will amplify only a subset of the restricted DNA fragments. Typically, a moderate to high number of bands will be observed, most of which may be monomorphic. So, polymorphic bands are identified as an RAPD analysis. AFLPs have typically been used to study variation among individuals of a species, most commonly for producing genetic maps and in trying to find genes responsible for certain traits. The power of AFLP is based upon the molecular genetic variations that exist within closely related species, varieties, or cultivars. The nucleic acid hybridization. Nucleic acid hybridization is a fundamental tool in molecular genetics which takes advantage of the ability of individual single-stranded nucleic acid molecules to form double-stranded molecules, that is, to hybridize to each other. For this to happen, the interacting single-stranded molecules must have a sufficient high degree of base complementarity. Nucleic acid hybridization is used to identify related or identical molecules. Nucleic acid hybridization assays involve a labeled nucleic acid probe to identify related DNA or RNA molecules present within a complex mixture of unlabeled nucleic acid molecules, the target nucleic acid. Nucleic acid hybrids can be formed between two strands of DNA, two strands of RNA, or one strand of DNA or one of RNA. The nucleic acid hybridization provides a means for detecting DNA or RNA sequences that are complementary to any isolated nucleic acid. So, nucleic acid hybridization, for this to happen, the interacting single-stranded molecules must have a sufficiently high degree of base complementarity. Nucleic acid hybridization is used to identify related or identical molecules. Nucleic acid hybridization assays involve a labeled nucleic acid probe to identify related DNA or RNA molecules present within a complex mixture of unlabeled nucleic acid molecules, the target nucleic acid. Nucleic acid hybrids can be formed between two strands of DNA, two strands of RNA, or one strand of DNA and one of RNA. Probe is required. Nucleic acid hybrids can be formed between two strands of DNA, two strands of RNA, and one strand of DNA and one of RNA. Nucleic acid hybridization involves a means for detecting DNA or RNA sequences that are complementary to the nucleic acid hybridization involves a means for detecting DNA or RNA sequences that are complementary to any isolated nucleic acid, such as a viral genome or a cloned DNA sequence. Nucleic acid hybridization can be used to detect homologous DNA or RNA sequences not only in the cell extracts but also in the chromosomes or intact cells, a procedure called in situ hybridization. In this case, the hybridization of radioactive or fluorescent props are specific cells or subcellular structures is analyzed by microscopic examination. For example, labeled props can be hybridized to labeled props can be hybridized to intact chromosomes in order to identify the chromosomal regions that contain a gene of interest. Then, stringency of a hybridization reaction. Stringency is a term that describes the number of mismatched bases that are allowed in a hybridization reaction and still have a double-stranded hybrid. The more stringent the hybridization conditions are, the fewer the mismatched bases that are allowed before the two strands come apart. Stringency is influenced by the temperature, the salt concentration of the hybridization mixture, and the pH of the hybridization reaction. All three of these conditions affect the stability of the double-stranded molecule. High stringency conditions are usually achieved with high temperature, approaching the melting temperature of the DNA hybrid, and low salt concentrations. High salt concentrations tend to stabilize the double stranded molecule and allow more mismatches to take place before the two strands come apart. The pH of the solution in which hybridization takes place also affects the stability of hybridizing molecule. Under slightly acidic conditions, pH 6 to 7, the double stranded DNA molecule is more stable because the increased H plus concentration also helps reduce the repulsive force between the two backbones of the DNA molecule. The nucleic acid probe. Nucleic acid used to carry a complex heterogeneous nucleic acid population as a hybridization assay is called a probe. Nucleic acid probes are segments of DNA or RNA that can bind with high specificity to complementary sequence of target DNA. Probes can be complementary to either RNA or DNA and can be from as few as 20 bases to many thousand of bases long. Nucleic acid probe may be DNA probes, RNA probes, or oligonucleotide probes. Then isotopic and non-isotopic labeling of nucleic acids. Isotopic labeling. Isotopic labeling of nucleic acid has been conducted by incorporating nucleotides containing radioisotopes. Such radio-labeled probes contain nucleotides within a radioisotope, often 32p, 33p, 35s, or tritium, which can be detected specifically in solution or much more commonly within a solid specimen. Autoradiography. The intensity of an autoradiographic signal is dependent on the intensity of the radiation emitted by the radioisotope and the time of expression, which may often be long, 1 or more days or even weeks in some applications. 32p has been used widely in southern blot hybridization, dot blot hybridization, colony and plague hybridization because it emits high energy beta particles which afford a high degree of sensitivity of detection. Next major topic is autoradiography. It is a technique of recording an image of a sample containing radioactive elements emitting radioactive particles or rays using photographic film, X-ray sensitive film, an emulsion, or other radiation sensitive medium. In autoradiography technique, samples are placed directly against the film for a period of time to allow radioactive emission from the sample to react with the film emission and create an image. A photographic emulsion is a suspension of a crystal of silver bromide embedded in gelatin, where when crystals of silver bromide are struck by charged particle or photon radiation, the silver atoms are ionized and form an invisible latent image. After exposure to the sample, the photographic grains in the emulsion are fixed using standard photographic developer which removes unionized silver bromide. After one emulsion is developed, each small aggregate of reduced silver atoms become a visible dark spot. Collectively, they make up the photographic image. Autoradiography has large number of practical applications in the biological, chemical, and physical science because it provides both qualitative and quantitative information. Then labeling DNA by Nick translation. Nick translation is a technique of radioactively labeled double-stranded DNA, making it suitable as a hybridization probe for detecting specific genomic sequences. It involves introduction of SS break nicks in the DNA. The internuclease DNA S1 is used to create nicks in a DNA probe fragment. Following DNA S1 treatment, DNA polymerase 1 is used to add nucleotide residues to the 3' hydroxyl ends created during the DNA S1 nicking process. As DNA polymerase 1 extends to 3' ends, the 5' to 3' exonuclease activity of the enzyme removes bases from the 5' phosphoryl terminus of the nick. The sequential addition of base onto the 3' end with simultaneous removal of bases from the 5' end of the downstream annealing strand results in translation of the nick along the DNA molecule. When performed in the presence of a radioactive deoxynucleoside triphosphate, such as alpha 32p DCTP, the newly synthesized strand becomes labeled. Nick translation is a technique for radioactively labeling DNA. or double-stranded DNA, making it suitable as a hybridization probe for detecting specific genomic sequence. It involves introduction of SS break nicks in the DNA. The endonuclease DNase 1 is used to create nicks in a DNA probe fragment. Following DNase 1 treatment, DNA polymerase 1 is used to add nucleotide residues to the free 3' hydroxyl lines created during the DNase 1 nicking process. As the DNA polymerase 1 extends the 3' ends, the 5' to 3' exonuclease activity of the enzyme removes bases from the 5' phosphoryl terminus of the nick. The sequential addition of base onto 3' end with the simultaneous removal of bases from the 5' end of the downstream annealing strand results in translation of the nick along the DNA molecule. When performed in the presence of radioactive deoxynucleoside triphosphate, the newly synthesized strand becomes labeled. DNase radio-labeled nucleotides. Translation DNA introduces single-stranded nicks by cleaving internal phosphodiester bonds, generating a 5' phosphate group and a 3' hydroxyl terminus. Addition of DNA pole 1 contributes to enzyme activities. A 5' 3' endonuclease attacks the exposed 5' terminus of nick and subsequently removes nucleotides in the 5' 3' direction, and the DNA polymerase adds new nucleotides to the exposed 3' hydroxyl group in the 5' 3' direction. In end labeling of DNA, single-stranded oligonucleotides are usually end labeled using polynucleotide kinase end labeling. Typically, the label is provided in the form of 3' 32P at the gamma phosphate position of ATP, and the polynucleotide kinase activity kinase catalyzes an exchange reaction within the 5' terminal phosphatase. The same procedure can also be used for labeling double-stranded DNA. In this case, fragments carrying label at one end only can then be generated by cleavage at an internal restriction site, generating two differentially sized fragments which can be separated by gel electrophoresis and purified. Then, non-anisotropic labeling. Non-anisotropic labeling systems involve the use of non-radioactive probes. Two types of non-radioactive labeling are conducted: direct non-isotropic labeling, where a nucleotide which contains the label that will be detected is incorporated. Often such systems involve incorporation of modified nucleotides containing a fluorophore, a chemical group which can fluoresce when exposed to light of a certain wavelength. Then indirect non-isotopic labeling, usually featuring the chemical coupling of a modified reporter molecule to a nucleotide precursor. After incorporation into DNA, the reporter groups can be usually featuring the chemical coupling of a modified reporter molecule to a nucleotide precursor. After incorporation into DNA, usually featuring the chemical coupling of a modified reporter molecule to a nucleotide precursor. After incorporation into DNA, the reporter groups can be specifically bound by an affinity group or molecule, a protein or other ligand which has a very high affinity for the reporter group. Conjugated to the later is a marker molecule or group which can be detected in a suitable assay. The reporter molecule or modified nucleotides need to protrude sufficiently far from the nucleic acid backbone to facilitate their detection by the affinity molecule, and so a long carbon atom spacer is required to separate the nucleotide from the reporter group. Then two indirect non-isotopic labeling systems are widely used. The biotin streptavidin system utilizes the extremely high affinity of two ligands, biotin, a naturally occurring vitamin, and acts as a reporter, and the bacterial protein streptavidin, which is the affinity molecule. Biotin and streptavidin bind together extremely tightly. Biotin and streptavidin bind together extremely tightly with an affinity constant of 10 raised to 14, one of the strongest known in biology. Biotinylated biotin and streptavidin. Biotinylated probes can be made easily by including a suitable biotinylated nucleotide in a labeling reaction. Then deoxygenin is a plant steroid obtained from digitalis plants to which a specific antibody has been raised. The deoxygenin specific antibody permits detection of nucleic acid molecules which have incorporated nucleotides containing the deoxygenin reporter group. Then bloating. Bloating is the transfer of nucleic acids or proteins from a gel strip to a specialized chemically reactive matrix called bloating membrane, typically nitrocellulose or activated nylon, on which the nucleic acids proteins may become covalently bound in a pattern similar to that present in the original gel. Then southern blot hybridization or southern blotting. Southern blot hybridization detects target DNA fragments that have been size fractioned by gel electrophoresis. This technique was invented in 1975 by E.M. Southern. In this procedure, the target DNA is digested with one or more restriction endonuclease, size fractioned by agarose gel electrophoresis, denatured and transferred to a nitrocellulose or nylon membrane for hybridization. During the electrophoresis, DNA fragments which are negatively charged because of the phosphate group are repelled from the negative electrode towards the positive electrode and sieved through the porous gel. Smaller DNA fragments move faster. Following electrophoresis, the test DNA fragments are denatured in strong alkali. As agarose gels are fragile and the DNA in them can diffuse within the gel, it is usual to transfer the denatured DNA fragments by bloating onto durable nitrocellulose or nylon membrane, to which single stranded DNA binds rapidly. The individual DNA fragments become immobilized on the membrane at positions which are a faithful record of the size separation achieved by agarose gel electrophoresis. Subsequently, the immobilized single stranded target DNA sequence are allowed to associate with labeled single stranded probe DNA. The probe will bind only to related sequences in the target DNA, and their position on the membrane can be related back to the original gel in order to estimate their size. Recombinant DNA technology and biotechnology. So DNA cleave with the restriction enzyme, gel electrophoresis, filter paper, alkaline solution, nitrocellulose gel, capillary action transfer DNA from gel to nitrocellulose. The Southern blot technique for detecting the presence of specific DNA sequences. The DNA is denatured and transferred from the gel to a nitrocellulose sheet by capillary action. Then northern blotting. Northern blotting or northern blot hybridization is a variant of southern blotting in which the target nucleic acid is RNA instead of DNA. In this technique, an RNA extract is electrophorated in a agarose gel using a denaturing buffer to ensure that RNAs do not form inter or intramolecular base pairs. After electrophoresis, the gel is bloated onto a nylon or nitrocellulose membrane and hybridized with the labeled probe. A principal use of this method is to obtain information on the expression patterns of specific genes. Next, Western blotting is a method by which proteins that have been separated and subsequently immobilized on the surface of a membrane are exposed and probed. When antibodies are used as probes, the procedure is called immunoblotting. Then dot blot assay. Dot blot assay, a specified volume of sample or specimen is spotted onto a small area of a nitrocellulose membrane. The spot is allowed to dry and is then treated with an alkaline solution to break open microbial cells and denature their DNA molecules. A solution of radioactively labeled probe is added to the membrane and allowed to react. After this, the membrane is washed several times to remove any unreacted probe and then allowed to dry. The membrane is covered with a photographic film that is sensitive to radioactivity, X-ray film, and then placed in a dark room. The film is developed and examined for the presence of dark spots. A dark spot indicates that the specimen contained DNA sequence that hybridized with the radioactive probe, and therefore that the sought target DNA is present in the specimen. If no dark spot appears on the film, it can be inferred that no DNA hybridization was occurred. The next major topic is DNA library or a genomic library, a library encompassing an entire genome. It involves cleavage of the entire genome of a cell with a specific restriction endonuclease and then cloning of each fragment, sometimes called the shotgun approach to gene cloning. This shotgun This technique can produce a very large number of DNA fragments on the order of a million to mammalian genome, which will generate millions of different colonies of transfected bacterial cells. Each of these colonies is composed of a clone of cells derived from a single ancestral cell and therefore harbors many copies of a particular stretch of the tough fragmented genome. Such a plasmid is said to contain a genomic DNA clone, and the entire collection of plasmids is called a genomic DNA library. But because the genomic DNA is cut into fragments at random, only some fragments contain genes. Many of the genomic DNA clones obtained from DNA of a higher eukaryotic cell contain only non-coding DNA. The presence of a particular gene is screened by a process of colony or in situ hybridization. Genomic DNA cleave with restriction enzymes, insert into vectors, host DNA, host cells take up DNA culture cells, a library of genomic clones. Many clones must be screened to obtain a gene of interest. A number of random cleavage fragments that must be cloned to ensure a high probability that a given sequence is represented at least once in the genomic library is calculated as follows. Probability P that some number of clones N contains a particular fragment representing a fraction F of the genome P 1 into 1 minus F raised to N.